Hello students, welcome to the SRBPS virtual classroom program. I am your biology educator Prerna Rathi. So students, in today's class we will be dealing with chapter 11 of your NCERT textbook, that's Biotechnology, Principles and Processes. So what are we going to learn in this chapter? We are going to learn the definition of biotechnology, the meaning basically of biotechnology, the various basic principles that are applied in biotechnology, the tools that are used in biotechnology, and the process of rDNA tech or the recombinant DNA technology. Let us first define biotechnology. Students, biotechnology deals with all the techniques of using live organisms, living organisms, or enzymes from those organisms to produce certain products and to devise certain methods or processes which are useful to humans. A better definition has been given by the EFB, that is the European Federation of Biotechnology. It says that biotechnology is the integration of natural science, that is biology, and organisms, cells, parts thereof, that means organisms and cells parts, and molecular analogues like proteins, nucleic acids, etc. for products and services. And once again, these products and services are for the mankind. The two basic principles that are implied in biotechnology are genetic engineering and the maintenance of sterile environment. What do you mean by genetic engineering? Genetic engineering is a technique by which we alter the DNA, we alter the genetic material, the chemistry of the genetic material, that is DNA or RNA, whatever is present, to introduce that into the host organism and thus change the phenotypic expression, that means to change the physical appearance or the expression of that particular organism. Because in biotechnology we are working with microorganisms, so we need to make sure that the environment that we are using is an absolutely sterile ambience and it is free of all sorts of infections, because the products that we are manufacturing here need to be really precise and accurate. The three basic steps that are used in order to genetically modify an organism is identification of the desirable DNA. जिस DNA में हमारा डिज़ायरेबल कैरेक्टर है, हमें टोटल DNA में से वो पर्टिकुलर जीन को आइडेंटिफाई करना है, उस आइडेंटिफाइड जीन को वहाँ से एक्सट्रैक्ट करना है, उसे होस्ट में इंट्रोड्यूस कराना है और उसे होस्ट में मेंटेन करना है, सो दैट दैट पर्टिकुलर कैरेक्टरिस्टिक इज ट्रांसफर्ड इनटू द होस्ट, into its progeny through that DNA. Various tools of biotechnology that we will be discussing today is restriction endonuclease enzymes, cloning vectors and a competent host. Let's discuss restriction enzymes. Restriction endonucleases क्या होते हैं? ये enzymes होते हैं which are also known as molecular scissors because they cut DNA molecules. The enzymes cut the DNA are known as restriction endonucleases. The first endonuclease that was found was HIND2. Let's see how this HIND2 has been named. H stands for Haemophilus, N stands for influenza, D strain second enzyme. That's how HIND2 has been named. EcoR1, that is E. coli R strain first enzyme. Okay, so that's how they have been named. Another thing that is very important about restriction endonucleases is the palindromic sequence. Palindromic sequence means a sequence that reads backward and forward same. For example, the word madam, MADAM, and backwards also MADAM. तो जितने भी restriction endonuclease होते हैं, इनका जो sequence होता है, जो recognition sequence होता है, वो हमेशा क्या होता है? palindromic होता है. यानी 5' to 3' जब हम उसको read करते हैं एक strand पे, तो वो same sound करता है जैसा वो 3' 5' वाले strand पे sound करता है. Understood? Now, why are these palindromic ends important? Because they create sticky ends. As you can see in this particular diagram here, first let us see the blunt end creation here, because they are not palindromic, so the ends that are created are blunt ends, whereas in case of this, read this GAATTC, and if you read it backwards, GAATTC, this is the sequence, so it reads the same backward and forward. So hence if we put a cut at G here and similar cut at G here, we will be having two sticky ends. Sticky ends are those complementary ends which are complementary to each other, which are ready to form the hydrogen bonds. Understood? Here again, once again, formation of sticky ends has been shown by color configuration. As you can see, the color coding is also perfect. It shows the clear formation of basically the complementary bonding, hydrogen bonding. The next technique that we have to learn about is gel electrophoresis. Gel electrophoresis, students, is a separation technique which is based on two basic principles. One, it is based on the sieving effect of the agarose gel. We use the agarose gel, which is D-galactose and 3,6-anhydro L-galactose mixture, in an electrolytic plate, and we connect that electrolytic plate to the electric supply. What happens after this? When the electrical supply is given, the DNA, being negatively charged, will start moving towards the positive terminal. When it moves towards the positive terminal, it is segregated according to its size, because the smaller pieces will move quickly and the larger pieces will not be able to move with that much speed. So the negative charge and the sieving effect of agarose gel are the two basic principles that are implied in gel electrophoresis technique. After they have been separated on the gel electrophoretic plate, a dye called ETBR, ethidium bromide, is applied. Then an imprint is taken on the nitrocellulose filter, which is then taken for autoradiography, where we come to know about the sequence. And the desirable sequence can be taken out of the gel by the process called elution. Fine. The next thing that we use, the tools that we use in biotechnology, are cloning vectors. Cloning vectors are used for preparing recombinant or rDNA. Fine. There are various things there are various properties that are necessary for in a cloning vector. It should have an origin of replication, or the ori gene. This ori gene is the place where replication will begin, and hence it would be able to create its copies. अगर ये अपनी ही कॉपी नहीं बना पाएगा, तो इसके अंदर जब आप डिज़ायरेबल DNA डाल दोगे तो ये उसकी कॉपी कैसे बना पाएगा? तो इसके अंदर ओरिजिन ऑफ रेप्लिकेशन का होना बहुत ज़्यादा ज़रूरी है. सेकेंड, सेलेक्टेबल मार्कर्स का होना. सेलेक्टेबल मार्कर्स वो मार्कर्स होते हैं जो हमें बाद में आफ्टर द एक्सपेरिमेंट ट्रांसफॉर्म्ड और नॉन ट्रांसफॉर्म्ड सेल्स को सेपरेट करने में हेल्प करते हैं. ये सेलेक्टेबल मार्कर्स रेजिस्टेंसेस हो सकती हैं, एंटीबायोटिक रेजिस्टेंस जैसे एम्पिसिलिन, टेट्रासाइक्लिन, क्लोरमफेनिकोल रेजिस्टेंसेस. Then cloning sites. Cloning sites are nothing but the recognition sites of various enzymes. ये भी होना बहुत ज़्यादा ज़रूरी है, तभी तो हम sticky ends create कर पाएंगे और तभी हम यहाँ पे desirable DNA को insert कर पाएंगे. Then the vectors for the cloning of plants and animals are separate. We can use yak and back vectors for this. Origin of replication. Sequence from where the replication starts is known as the ori. Any piece of DNA, अगर हम किसी भी piece को इस ori के साथ relate कर देते हैं तो वो भी अपना copy number increase करेगा, वो बार बार replicate करेगा. Hence this origin of replication is very important. Next, selectable markers. I told you they are helpful in segregating the transformants and non-transformants. Then cloning sites, as you can see, various. This is the diagram of PBR322, which is an important enzyme. It has sites PST1, PVU1, EcoR1, CLAV1, HIND3, BAMH1, SAL1, PVU1. It has origin of replication and the proteins responsible for replication, ampicillin resistance and tetracycline. So this is basically a complete cloning vector which has everything. It has drug resistances which act as the selectable markers, it has origin of replication, and it has multiple cloning sites as well. Fine. Selection of recombinant is done by insertional inactivation of the removed gene due to the new insert. जो हम न्यू इंसर्ट डालते हैं वो हम किस में डाल देंगे? एंटीबायोटिक रेजिस्टेंस में डाल देंगे. तो उससे क्या हो जाएगा? इंसर्शनल इनएक्टिवेशन हो जाएगा. अब जब आप उसको सपोज आपने एम्पिसिलिन रेजिस्टेंस को डीएक्टिवेट कर दिया, तो अगर आप उसको एम्पिसिलिन पे कल्चर करके देखोगे तो वो कल्चर नहीं होगा. दिस शोज दैट इट हैज़ ट्रांसफॉर्म्ड. अगर वो ट्रांसफॉर्म हो जाएगा तो एम्पिसिलिन पे कल्चर नहीं होगा. अगर वो ट्रांसफॉर्म होगा, अगर नहीं हुआ होगा ट्रांसफॉर्म, तभी वो एम्पिसिलिन पे कल्चर होगा. एंड लाइक दिस वी कैन क्रिएट द जीन लाइब्रेरीज फॉर वेरियस DNA samples. Then what are cloning vectors? Cloning vectors are anything that will take the desirable DNA, the recombinant DNA, into the host. For example, agrobacterium mediated transformation we can use in plants, okay, where we use the TI plasmid which is present in the tDNA. The Ti plasmid is there. This is the Ti plasmid, as you can say. It has a tumor inducing DNA. हम क्या करते हैं, इसकी जो tumor inducing capacity है उसको silent कर देते हैं by inserting the desirable DNA in the tDNA. तो अब ये ट्यूमर तो इंड्यूस नहीं कर पाएगा, अभी डिजीज़ तो कॉज नहीं कर पाएगा. अदरवाइज एग्रोबैक्टीरियम ट्यूमरफिशंस एक डिजीज़ कॉजिंग पैथोजन था. ये क्या कर रहा था, डिजीज़ कॉज कर रहा था प्लांट्स में. अभी डिजीज़ नहीं कॉज कर पाएगा, लेकिन क्योंकि इसका रेट ऑफ इन्फेक्शन बहुत हाई है, ये होस्ट के अंदर डिज़ायरेबल डीएनए को डिलीवर कर देगा. अंडरस्टूड एवरीवन? As you can see here, non-viral gene delivery is there. Then you can use adenoviruses and retroviruses in case of animals. Okay, so these are various methods that we can use in case of animals and plants. We'll be discussing further in our next class. I hope you all understood what I taught. I suggest you all make complete use of the YouTube channel to enhance your learning. So until next time, goodbye, take care.